Hello everyone. Welcome back to Microbial Engineering and Algal Biotechnology Laboratory YouTube channel. And today we are going to discuss a very interesting topic on bioinformatics tools for metabolic engineering and synthetic biology. So this is a glimpse that has been taken from an invited talk on faculty development program, where I got an invitation and delivered my lecture at NIT Warangal in 2021. So let's get started the topic on the bioinformatic tools for metabolic engineering and synthetic biology. I hope this particular lecture session will help our student a lot, because lots of new advanced technologies has been depicted here as a summarized form. So if you need to go for further studies, I have included different references over here, so you can go on through and get enrich and nurture your knowledge further. So what is metabolic engineering? Metabolic engineering is the practice of optimizing genetic and regulatory processes like anabolic as well as the catabolic enzymatic networks within the cell or cell factories to increase or ameliorate the cell's production on the biosynthetic ability to certain substances. So the substances which are referred to as the value-added products. Then how you can able to define the synthetic biology abbreviation. Synthetic biology is a multidisciplinary area of research that seeks to create and generate new biological compartments or the parts, like microbial cell factory device developments. And that will allow us also to come up with some systems or to redesign the systems that are already found in nature or semi-synthetic in nature as well. Now, this is a very interesting scenario, like systems biology. Now, when you have been considering the metabolic engineering and system synthetics biology, that will allow to make a model, and that will be called the system biology. So system biology is a computational and mathematical analysis and modeling of the complex biological systems or network to optimize and simulate the efficiency of microbial cell factories, like if you consider metabolically engineered and or incorporated synthetic biological circuits via holistic approach, because lots of algorithm and programs are there that will allow you to develop the models as a system to study the interactive impacts on the biochemical, physicochemical reactions within the biological system. Even you can also able to evaluate how this system or model organism can react when an external or internal signals will be applied or the organism will get exposed into it. So this is the synergistic pictorial presentation. It shows that how the systems biology, synthetic biology, and metabolic engineering, they are orchestrated manner operating in a parallel way. So systems biology, you can see this is kind of mathematical model. Like in systems biology, you can use computational tool to design for forward engineering of genetic circuits, different biological segments. If you consider operator sequence, the promoter sequence, the structural or functional genes, even the regulatory sequences which are present in the upstream truncated regions or the downstream terminator sequences as well. So synthetic biology will allow you to design the synthetic compartments to reorient and rearrange in different ways that you can alter the gene expression and also level in the transcription as well as in the translational level. Whereas metabolic engineering, it's a kind of computational analysis that will allow to simulate and balance and equilibrate the metabolic networks, because in the metabolic network, a lot of enzymatic reactions are always got involved. Now, if all the enzymes, they do function in an orchestrated, equilibrated manner, unless and until the enzyme metabolic pathway may not be properly functioning to give rise you a certain product that you are expected to. Now, the synthetic biology and metabolic engineering, when you have been put together and you can able to come up with a mathematical expression out of it to explain the entire biological platform or the arena, how it will be get regulated when it will get some exposure to the external or internal stress factors and how they do react, how the alteration will happen. That can easily be explained by a development of model, especially the mathematical statistical model that will allow you to get a complete overview that will be only get by using or applying the system biology approach. So these are the synergy between the three basic terminology that is the main focus of our discussion today. So this is also a nice pictorial presentation which shows that how systems biology synthetic biology and computational science, they are co-merging with each other. The systems biology that will allow you to understand the natural gene networks, whereas the computational science will allow you to understand and allow you to design the modeling and analyzing data set, whereas metabolic engineering will allow to build up new gene network. And ultimately, synthetic biology will come and put forward all these things together to make a microbial cell factory to get functionalized. So this is the concept of microbial cell factory, because I have been talking several terms on the microbial cell factory. So let's talk about what is the concept or lies behind the microbial cell factory. So microbial cell factory is an approach for bioengineering which considers the microbial cells as a production facility, in which the optimization of the process largely depends on the metabolic engineering. Microbial cell factories are also being utilized for many applications in industrial microbiology, even in the medical sectors. You can see nice pictorial presentation have been shown that how the synthetic pathway optimization and also optimization of the growth of certain microbial cell factory will allow you to give maximum substrate conversion efficiency and allow you to give rise to maximum product yield or the productivity along with the side products. So this is an orchestrated manner which is being happening inside the microorganism. So you can able to optimize and you can also able to simulate the microorganism in such a way that it can give you the maximum conversion of substrate to your desired products. So microbial cell factory is an approach for bioengineering, which considers the microbial cells as a production facility in which the optimization of the process means the different enzymatic reactions largely depends on the metabolic engineering, and microbial cell factories are used for many applications in medical sciences as well. So this is the overall concept of microbial cell factory, how you can convert a microorganism which is low-yielding microorganism and you can transform them into a high-yielding microbial community. Like if you have considered E. coli as a native microorganism, when you have been collecting from the nature, you have been isolating from the nature, what you can find, the productivity that you have been expecting or the expectation of the industrial large-scale production, that cannot be attained. Now, when you have been applying different approaches related to metabolic engineering, modification of the related pathways, knocking out or knocking down of your competing pathways, that will allow you to ultimately give rise to an engineered E. coli cell which having higher yield or molar productivity. So what are the basic steps which has to be considered while you have been considering to engineer a microorganism or naive microorganism to transform them into microbial potential cell factory? So number one step would be the transportation, the influx of the substrate that you are looking into, convert them into precursor, intermediates, and finally to give rise a product. Finally, the glucose when get inside or the carbon source will get inside the cell, then central metabolic pathway needs to be get activated. And finally, different amino acid pathway can also be considered during the alterations while you have been considering the product improvement. Similar way, you can also knock down or knock out the branch pathway. These are called the competitive pathways. And finally, you can also able to break down. You can also introduce some degradation pathway which will allow to break down some toxic metabolites. And finally, the transport pathway should get induced because the product that have been generating inside the cytosol or within the microorganism that needs to get out, that easily can get separated from the cellular biomass. So that is called the PTA-ACK pathway, or somehow get related with the efflux transport pathway. So this is the overall schema to conceptualize the basic outline on the microbial cell factory development and its proper equilibrations to improve the desired product yield. So this is another pictorial presentation which shows out how the substrate can get in. That will be the rate controlling step, and there are lots of enzymes which are being involved, which will help to carry out the pathway balance. The pathway balance can also be considered different subcellular downstream processings of the pathways like precursor or the cofactor supply, competing pathway alterations, cofactor regeneration system, like if you consider some oxygenase reaction, some oxidoredox reaction, some redox reaction, any kind of reduction reaction, if you can consider, then you have to maintain the proper proportion of different redox equivalents. That means electron carriers. These are none other than the NADH, FADH2, FMN. These are responsible. So their regeneration is also very much important, because unless you recycle them back into their oxidized or the reduced form, you cannot able to carry out the pathway for your downstream product generation. Similar way, you have to carry out also another important scenario, that is the chassis optimization. Chassis means the host optimization. That means whether your host can able to tolerate the product that you have been expecting in a certain titer as per the stoichiometric equation. So this is the concept that we have been following in the next slide, that is the concept of systems metabolic engineering. So it is a nice pictorial presentation which shows a very interesting triad. The number one part of this triad is the pathway fused approaches. Second one is the evolutionary approaches, and the third segment is the systems biology based approaches. So the strategy which are involved in the pathway fused approaches, those are includes the carbon source utilization, precursor enrichment, byproduct elimination, degradation, blocking, transporter engineering, and also recycling or the regeneration of the different redox cofactors. Finally, the next better important segment is the evolutionary approach. Like the strategies are the enzyme evolution, metabolic evolution, adaptive evolution. There are several techniques which are under the application, like FACS or the biosensor, which will allow you to understand those facts and phenomena. The final segment of this system metabolic engineering triad concept is the systems biology based approaches. These are mostly based on the omics based approaches, includes genomics, transcriptomics, proteomics, metabolomics, fluxomics. Even nowadays a new terminology has come up that is called the materialomics. So all these things has been done in the in silico simulation part using the computer algorithms, and then you can carry out the validation experiment. Finally, to make sure like the cell factory that you have been designed to get functionalized, to get the higher conversion of substrate to your desired product is fully functional or not, or it is balanced or not. It can reduce the cost of overall industrial bioprocess industry or not. These kind of things you have to cross-check by the validation experimentation as a follow-up study. So these are the very, very complicated structure, I would say. This has been taken from a Nature paper. It shows then an overall strategy and procedures which are involved in systems metabolic engineering for the bio-based production of the chemical or the materials. So this is another concept that has been associated with the concept of strain engineering, because we have been talking several times about the host engineering, the host chassis engineering. So these are none other than the engineering of the host, where the metabolic pathway or synthetic circuit will be get functionalized, either overexpressed or either the control of the toggle switching mechanism. So that will be based on the two basic aspects or the salient features. Number one, that is the product selection. That will entirely depends on the initial concentration of the substrate that you have been chosen and the development of the intermediate precursors and final product outcome. So these are mostly depending on the chemical and biological conversion based on the bulk chemicals, fine chemicals, polymer or materials, biofuels or the natural products. On the similar end, on the other end of the coin is the host strain selection. So suppose you have been trying to develop a specific bioproduct like any kind of alkane production, if you consider as an example. Now, if you have been chosen a host which can't able to tolerate the hydrocarbon in a higher level or above the threshold level, then you should not choose that particular strain, because strain is not at all suitable to survive when your non-natural product has been synthesized and generated within the cell. That can be toxic to the cell. That's why host selection plays a very important role that will allow you to understand the host will really be a good chassis that will allow or that can able to sustain the toxic impacts of the product that you have been expecting to get biosynthesized within that chassis. So these are other two aspects that you have to always get consideration while you have been performing the chassis engineering, that is the pathway construction. Because lots of metabolic pathways which are naively existing within this strain that can also compare with the precursor availability or the accumulation of the toxic metabolites. So that is called the pathway construction, plays very important role while you have been choosing any particular chassis or the strain. That has to be get engineered by knocking out or knocking down the competing pathway, not to get accumulation of the precursor or the toxic intermediate that can easily get flux towards the final products. So That's why the final step has come forward to optimize or to design the strain, that is the evolutionary engineering. So this is none other than the random library or the interactive evolution. Sometimes we would also say this is an adaptive laboratory evolution that will direct or force the particular strain that they can attain or achieve certain tolerance against the product that you have been trying to biosynthesize within that strain, which is not naturally able to tolerate that product, which is completely non-natural to that particular host strain. So these are the summarization, how you can able to carry out the optimization of the metabolic NAIP pathway or the synthetic pathway. So there are the basic 4 terminologies are there involved. Number 1, these are the avenues through which you can able to carry out the pathway optimization during the strain engineering. Number 1, that is the genome scale metabolic model development. So that is completely algorithm and computational based approach that has to be get validated by experimental results after simulating your entire pathway that have already been functionalized within the suitable chassis strain. Next one is the plasmid based engineering. Suppose whether you have been looking to express your circuit in an extra chromosomal chassis or you wanted to get integrated within the host genome or the host chromosome. Third one is the regulatory RNA-based engineering. Suppose you want to get some kind of switch on and switch off mechanism that do exist in the viral system. So you can introduce this kind of regulatory RNA-based engineering approaches within your chassis strain. And finally, the fourth one is the genome-based engineering by using knocking out certain gene expression, either by using lambda red recombinase system or by using the CRISPR-Cas9 system that is the current use. People have been using a lot in different regime of microbial communities at current scenario or at the current days. Now let's talk about the biorefinery approach. That is very important because whenever you have come up with your chassis strain is ready, that can have the higher substrate conversion efficiency and highest yielding strain you have already gained after the simulation and validation, then you have to think how you can able to functionalize this microorganism to get used in the large scale production. So that will be entirely lies on the concept of biorefinery approach. A biorefinery approach which involves 4 different processes. Number 1, the first upstream process that converts the renewable biomass into fermentable carbohydrates. Second, upstream process that develops a microbial strain that do capable of efficiently producing a desired product. Third one is a midstream process that cultivates the microbial communities and produce a chemical or material of interest, that is your desired product or target. And finally, the downstream processing that recovers, purifies the desired product. It's very simple. Like if your productivity is quite high, you automatically drops down the downstream processing cost, and that will be really sustainable and feasible process as per the concept of biorefinery approach to get carried out, to get mimic in the large scale production. That will always become very, very beneficial to get applied in the industrial sectors. Although none of these 4 processes is uniquely important, the second upstream process of developing microbial strain is the most primordial as it determines the overall efficacy of converting raw materials, which is a cheaper feedstock, to a value-added product. Since microbial community have been isolated from nature, are not always optimized for the production of our desired product. It is quite natural and normal. Their performance needs to be get enhanced or ameliorated. This is where metabolic engineering, system biology, and synthetic metabolic circuit comes into play. Metabolic engineering can be defined as a purposeful modification or alterations of a cellular network to attain or achieve defined objective. That is our expectation. So these are the basic trends of the systems synthetic metabolic engineering as per their chronology of discovery, and the trend is moving forward. So it has been get started in 1990s, and it has been now come to a dynamic strain designing phase and synthetic eukaryotic genome development, even ribosome binding calculation, expression matrix, co-culture modeling, knock-off design lactic acid E. coli. These are the different techniques has been come up time to time. That has been the chronology that has been started from 90s, and now it is come to an 21st century with more advanced and technologically sound approaches. So then what are the basic rationale behind it? Why we should try to use some bioinformatics tools to make a faster or rapid progression in the field of metabolic systems biology. So bioinformatic tools are the building blocks for pursuing systems, metabolic engineering, and synthetic biology approaches to successfully construct potential microbial cell factories to establish and attain sustainable biorefinery platform towards the value-added biomaterial generation. So that is the basic rationale behind why we should merge the bioinformatic online or offline tools or the computational algorithm and programs. And that will play a very, very primordial role in the field of metabolic engineering and synthetic biological sciences advancements. Now, what are the computational tools which are playing very important role in the field of systems metabolic engineering and synthetic biology? What are the basic purpose are to be governed? Number one, the reconstructing metabolic network. So metabolic network reconstructions describe the identity and functions of the constituents, those who are involved in an organism's metabolism, by linking metabolic and genomic or even transcriptomics or proteomics, metabolomics, fluxomics dataset. Secondly, the visualization of metabolic networks that you have been designed. So network visualization will allow the researcher to leverage the power of human intuition to identify or fetch out the patterns that are undetectable by computational algorithms. Network visualization serves as a bridge that links metabolic network reconstructions to downstream analysis and also further design methods. So these are the basic computational tools which are the part of Bioinformatics that will allow you to reconstructing the metabolic network. So these includes mostly BLAST, CAS, CAKE, model seed, sequence ontology, metasys, pathway tools, biochemical, genetics and genome knowledge bases, exchange language, seed framework, the pathologics, the biosys, gene ontology, CAKE pathway, CAKE translator, MET reaction, systems biology ontology. So these are the most important algorithms which you can apply to reconstruct your metabolic network. And number two is the visualization of this network. So what are those computational tools that you can apply to visualize the metabolic network? Those were includes the cytoscape, the SynStreet, the GRAM, the Acrodia, Omics, Cell Designer, Tinkler Systems Biology Graphical Notation, GraphViz, the Cytoseed, Vented, CellNet Analyzer, Cell Illustrator, Biochemistry, and also Synthetic Biology Open Language. These are mostly algorithms that you can try to implement in your system that will allow you to simulate your cell factory in the in silico approach. Definitely, you have to validate it by carrying out the experimental setup and also do lots of molecular biology work that lies behind it. Third one is the manipulating biological molecules. Ultimately, implementing alterations in the metabolic network requires the direct manipulation of the biological molecules that do synthesizing the nucleic acid, protein, constructing expression cascade, mutating nucleic acid sequences are very common in metabolic engineering tasks that involve the physical modifications of the biological modules. Fourth, which is very, very important, that is the metabolic flux analysis. So maximal cellular production of an interesting compound is often the defining goal of metabolic engineering. Strategies that roots out the cellular resources away from the non-essential pathways in an organism are more likely to result in the desired production goal. Now let's talk about and let's have a visualized form how you can able to use bioinformatic tools or computational algorithm that will allow you to manipulate the biological molecules. Those who are including the tickler cells, the Genius, the DNA star, laser gene, the DNA works, the NewPack, RBS calculator, GenoCAD, Vector NTI, Plasmid editor, Fuchsia, Vienna RNA, Web Suite, and Gene Designer. Now, fourth one is how you can able to carry out the metabolic flux analysis by using different ranges of bioinformatic tools. So the bioinformatic tools that will allow you to calculate the metabolic flux analysis, those who are including the FBA, Room, OptFlux, CellNet Designer, System Biology Research Toolbox, the Fate Flux, MoMA, Cobra Toolbox, OptNoc, Facimo, OpenFlux platform for MATLAB. So these are mostly very complicated kind of algorithms and software. You definitely need to have some kind of programming idea, especially on C programming, C++, and also Python, little bit of Perl, and also visual inspection of this. So these are the mostly the computational biologists, they do carry out this kind of work and developing the big data set. They allow you to understand and calculate and simulate the metabolic flux analysis. Now the fourth and fifth one is very, very co-merged. So fifth one is the pathway prospecting and the permissive host selection. So this approach involves the developing techniques to effectively search for identifying the exogenous reaction, metabolic pathways, suitable metabolically competent cells, mostly host microbial model system that can be introduced into the standard production system. So this is actually called the permissive host development. Sixth point, dealing with the post-structural network analysis. It deals with the understanding the design of metabolic network. Locating enzyme targets is to determine the most likely enzyme target considering the thermodynamic constant, because lots of rules and regulations are there that will allow to establish the thermodynamic constant for altering the expression level in the transcription or in the translation, post-translational modification. Allow you to understand the stability and the quantity of the messenger RNA transcript level and their stability, and also the stability of protein and level of expression. So what are the computational and bioinformatic tools are available that will allow you to prospecting the pathway and also allow to identify a permissive host? So these are mostly three softwares are available. Mostly these are computational tools: integrated microbial genome, the OpStrain and OpNoc. And finally, the post-structural network analysis can also be done by utilizing certain algorithms on COPACI, systems biology workbench, the PHYSAs and Tinkler cells. Now the seventh point is very, very important, how you can able to optimize the microbial culture. So introducing design pathway or the foreign or the named pathway into a host for heterogeneous expression often results in poor or no growth at all for a variety of reasons. These reasons are including the non-suitable host selection or the low substrate availability or a shortage or accumulation of intermediate toxic metabolites, also upregulation of the competing metabolic pathways, or restricting storage capacity of the end product. So culture optimization or CO that will help to use the metabolic model with the primary intention of maximizing the growth rate or the growth yield or product yield for performing the flux balance analysis or the FBA platform. So since altering metabolism may slow down the growth, or metabolic engineer need to consider the effect of the culture conditions, that is the internal or external microenvironment, on maximum product yield development. Considering every variables related to culture conditions is daunting. However, certain tools can help optimize growth and direct the flux towards a product of interest. Most of these tools assess growth under various cultural conditions by applying the flux analysis methods to genome scan metabolic models with the primary intention of maximizing the growth rate, as discussed earlier during the FBA platform can be used towards this end. Now, these are the basic softwares or the computational algorithm that have been applied to carry out the optimization of culture and pathway, like GrowMatch or the BioInformatic Toolbox. These are not very simple to operate. You have to have a complete knowledge about the computational programming as such. The eighth one is the toxic intermediate optimization. It involves the considering toxic intermediates during pathway optimization. It accelerates to help to perform the statistical analysis to carry out the significance as per the statistical concern on the metabolomics dataset in an effort to link the phenotypes with the metabolites level. So these are the certain very limited softwares are available that will allow to carry out the toxic intermediate optimization, including the MetaP server and the MELT database, that is MELT-DB. Now, what should be the target? Now, whatever you have learned so far, how you can able to target a molecule. That means the target-oriented workflow one that I have been discussing. So this paper I have been published in 2017, that is System Biology PAVE's Pathway and Potential Enzyme Prediction towards Anti-Cancer Drug Methyl Jasponate Biosynthesis. So this is the semantic diagram of the biosynthetic pathway for methyl jasponate or jasponic acid in the model plant Arabidopsis thaliana. So this is natural or the naive pathway which are present in this particular plant model. But this is very complicated model. You cannot able to functionalize in the microbial platform. That's why you have to do some kind of plug and play to make it much more simpler and that you can easily handle within the prokaryotic platform. So to do that, I have designed different microbial based algorithms and programming, and this is the workflow how it is get executed. So that will allow you to understand how this system biology workflow will allow towards the pathway prediction and the designing of this pathway in a shorter form. So you have to put the query sequence over there, then it will carry out the program that I have developed that will carry out the auto execution inside. That will also consider different metabolic constraint, thermodynamic constraint, biochemical constraint, that will execute it all together in parallel. And ultimately you can able to come up with the output. So output will be includes with the pathway ranking. That means it will give you 10 pathways, design pathways, but maybe 1 or 2 will be very good. So that will based on the ranking 1, 2, and 3 and so forth. Pathway information would be there, and also the reaction information will also be included, like what should be the free energy change, what would be the entropy change, what will be the enthalpy change of this entire metabolic set of reaction that has been designed by using my algorithm that I have been developing. So based on that algorithm that we have used, this is the predicted biosynthetic pathway design for the stearic acid to oleic acid conversion towards, because stearic acid is the substrate and oleic acid is the intermediate precursor. Now this oleic acid will further be converted into the final product, that is methyl jasmonate. So these are the, you can see this diagram will clearly shows that this particular programming that I have been developing or I have developed already, that will also give you the individual reaction steps, also including different enzyme classes are also being included. It will also allow you to give rise the free energy change that will allow you to understand whether this reaction is energetically or entropically favorable or not. That means if you want to functionalize this synthetic rank pathway, it will allow you to carry out the flux towards methyl jasmonate from stearic acid or not. So that is the thing that I have already shown on this slide, which is one example of target-oriented workflow. So this program will not only give you the information about the enzyme commission, it will also give you the information about individual enzymes which are belong to that enzyme commission class. So that will allow you to give you information about the nucleotide or the protein sequence regarding those particular enzyme which belongs to that particular class of enzyme, which is involved in the conversion from individual steps within this entire metabolic network that have been designed by using my own algorithm and software applications. This is another target-oriented workflow too. This paper also been published in 2017, where I have been used this particular algorithm and program to understand and identify certain novel enzymes that will allow to biosynthesize the cholesterol-lowering drug. And that cholesterol-lowering drug has been derived from a crude waste material of the crude glycerol. So this is the similar way. This is the natural pathway which is existing within. This is called the BTO biosynthetic pathway, which is present in E. coli as well as in Arabidopsis thaliana. So this is the NAE pathway, but the pathway is very complicated to get executed within the microbial system because lots of regulation needs to be get involved, and that is not possible to functionalize in the model microorganism E. coli or any other class of gram-positive microorganisms over there. So this is the system biology tool. This is kind of web-based tool I have designed as a workflow for prediction and design of biosynthetic pathways. And this is also allow you to understand how the glycerol can be converted into the 1,2,4-butane triol, that is called BTO. So this is the predicted and designed putative synthetic metabolic network concerning the glycerol entry node to the BTO biosynthesis, including thermodynamic constraint and enzyme classes, based on the free energy change data set and how it will vary, how it will allow you to understand based on the kilocalorie per mole unit based on the free energy change. So that will allow you to understand whether this workflow or the individual enzymatic steps will be energetically or thermodynamically favorable or not. So that kind of data can also be, you will get by applying this algorithm or software. So these are the specific references that I have been chosen while I have been delivering this particular kind of lecture session. I hope you can go for further reading, because these researchers are the very eminent researchers. Some of them are the father of this metabolic engineering concept, like Edward or the Paulson. also Gregor Stephanopoulos, they are the fathers. So I had a very great opportunity to get the opportunity to meet them and also discuss with lots of my work that I have been performing in a collaboration. So that's all from my end. Now, if you have any kind of question, any comments, if you want to know something, if you want to clear your doubts further, please you can put your comments in the YouTube chat box. I will be happy to answer time to time when I will get the spare time. So thank you very much. I hope you enjoyed this session. You have learned a lot of things and you get motivated to learn more on this particular topic. So thank you very much.